Hi, everyone. Welcome to Biochemistry 2. And we are going to start off on our very first pathway, which is glycolysis. Before stepping into kind of our pathways individually, I want to kind of give you a big picture analysis of metabolism, which is the main topic that we're going to be looking at in biochemistry 2. Often we see these very complex network maps, as shown here, as representations of metabolism, and it can get quite confusing and quite complex. This might look like some crazy metro map to some of you. But what we're going to be doing in this class is breaking it down and individually looking at the contributions of each main pathway within this whole map and some of its relations to other pathways. I want to note that this class is going to be broken up into what I would say is two main sections. One section is going to be all about pathways where we're looking at how molecules get broken down by the body. Another word for this is catabolism. The other half of the class is going to be looking at the opposite. How do molecules get built up or pieced together into something larger in the body? This is also called anabolism. So, two sections. First, we're going to be looking at a bunch of breakdown pathways, catabolic, and then we'll be looking at a lot of build up pathways, anabolic. So here's kind of a close-up on our first pathway we're going to be looking at, glycolysis, which is a catabolic process. You can actually see I've zoomed into this pathway, and you can start to see where glycolysis is. It's very small relative to that large map I just showed you, but it is one of the most important and key pathways in most organisms, most living organisms in general. Now, if you get any interest in looking into these pathways further and where I got that map in general, I've actually put a link here. You can go to kegg.com, that's KEGG.com, or just Google search KEGG in general. It has all of the metabolic pathways that we're going to be talking about. It's where you can see this big map that I got, and any pathway that we start to go into, you can look at in more detail on KEGG. I think KEGG is a great resource, not just for this class, outside of this class as well, to kind of understand metabolism and nutrition. So the topics of this first section, we're going to be talking about how organisms, including us as humans, harness energy from glucose, and that is via this pathway called glycolysis. We're also going to be touching into another type of related tangential pathway called fermentation, and this is going to be under anaerobic conditions. So this is conditions in which we have low oxygen. So our first step is to talk about glucose. We've looked at glucose in the past. It is a carbohydrate and it is an excellent fuel. The reason that glucose is important is because it yields a lot of energy when it is oxidized. It can also be stored efficiently in polymeric form. We talked about this again when we looked at general carbohydrates. We looked at all of its types of starches, which are the polymeric forms of glucose. So we can store it very easily and we can break off pieces when needed. So many organisms out there, and also many types of tissues within organisms, can actually meet all of their energy needs just using glucose. It's also very versatile. We've talked about how carbohydrates can be used as energy, but also be used as building blocks. If you remember, bacteria in particular can actually use glucose and other carbohydrates for building blocks in their carbon skeletons. So, amino acids, membrane lipids, nucleotides in DNA and RNA, and all of the cofactors needed for metabolism, all of those carbon atoms that we need in those molecules can be utilized, can be gathered from breaking glucose down first. Glucose has a lot of carbon in it, and then building up those molecules. So, what are some pathways where glucose is utilized? Again, as mentioned, glucose can be stored. There are storage pathways. It can be stored in the polymeric forms of starch and glycogen. And this is going to happen when we have plenty of excess energy, right? You don't need to be breaking down glucose for fuel. And so it can then be shuttled off to be stored. There is, of course, glycolysis, which we will be talking about, the breakdown of glucose, which generates energy via the oxidation of glucose. And this is going to be when we have short term needs of energy. Glucose utilization also feeds into the pentose phosphate pathway, which we will be touching on in the future, which is going to generate this molecule NADPH. Again, this is generated via the oxidation of glucose. And so it feeds into the pathway this way via NADPH. And this is utilized actually in lipids and nucleotides. So it's used for detoxification and biosynthesis of lipids and nucleotides. So glucose is still tangentially related to lipids and nucleotides. Glucose can also be used for structural components. So structural polysaccharides, for example, can exist in cell walls. Particularly, this is going to be seen in bacteria and fungi and plants. So all of these organisms actually utilize sugars on their outside of their cell walls. Glycolysis is a 10 series reaction, and it takes place in the cytosol of the cell. This is an important key thing to note. And through the semester, I want you to be thinking about and remembering where these pathways take place. It seems trivial now, but it's actually very important. Cells are inherently compartmentalized so that important pathways can take place in certain areas. And those compartments are actually built to get the most out of those particular pathways. So, thinking about where this happens is important. So, again, glycolysis happens in the cytosol of the cell. Remember, that's kind of the outer component of the cell. There's no other specific organelle that exists that is compartmentalizing glycolysis except for the cell itself. And again, the point is to extract energy from glucose. And we're going to do this by splitting glucose into two 3-carbon molecules called pyruvate. Glucose is a 6-carbon molecule, and it's going to be split into 2 pyruvates at the very, very end. That's our end goal. This pyruvate is, as you might have guessed, going to continue into other pathways because glycolysis is linked to so many different pathways. It can be further oxidized to get more energy out of, or it can be used to build things up. So via the catabolism, the breakdown of glucose, then its products can be sent off to do a variety of things. We can have further breakdown, or we can have building blocks that build things. Um, and this is going to be just dependent on what the needs are of the organism. This pathway, the reason we start with this pathway and we talk about this pathway first is because number one, it's connected to so many other pathways. But number two, it's actually a very ancient metabolic pathway. What I mean by that is it's one of the first evolved pathways that organisms started to use. It's the first stage of cellular oxidation. So, at a cellular level, this is the first stage in which we start to oxidize products. And the key thing about glycolysis is that it doesn't require oxygen. So you can see this is connected to its ancient evolution. Before we had oxygen-rich environments, there needed to be organisms that could power themselves via breakdown of some product without using oxygen. Now, this breakdown in glycolysis is then captured via the synthesis of some other molecules we're going to be talking a lot about in this semester. These are carrier molecules, which we'll go into more, called ATP and NADH, both of which are energy carrying molecules. These are like the currency of the body. And again, we're going to be talking about them a whole lot and their currency and which pathways give us how much. We're going to be talking about them like they're money, right? Like, how much money do I get out of glycolysis? How much currency do I get out of this? And these are our currency. Now, I know I started this conversation talking about glucose, but I want to be clear that many carbohydrates besides glucose can actually lead into glycolysis. So, most sugars that you consume eventually are going to meet their breakdown fate in the glycolysis pathway. And they do this once they are transformed into one of the intermediates in the pathway. I said this is a 10-step pathway. That means there are 10 individual intermediate products along the pathway. And different sugars can be transformed into any of those points or one of those points along the pathway. And you can see here, for example, galactose. Galactose is a sugar that you actually find in milk. It's part of the lactose dimer. And galactose can be broken down and then altered to form this glucose 1 phosphate, which is part of the main pathway of glycolysis. You can see that fructose, which is part of sucrose, can be transformed into this fructose 6 phosphate, which is again part of the main pathway of glycolysis. So generally, the most significant sugars that that are going to be utilized in this sense are storage polysaccharides, so glycogen and starch. Those are the obvious ones that are going to utilize glucose to further go into glycolysis, or sugars that we get from our diet, right? So these ones I just talked about, like fructose and galactose, come from our diet. They're from disaccharides. So go back and remember, disaccharides are carbohydrates made of two sugars, right? Some of the main disaccharides that we see, sucrose, lactose, right? We get that from dietary in taking in dairy, right? Sucrose is table sugar. So we see sucrose all the time because we consume sucrose. Or maltose. Maltose also is a sugar that is consumed semi-often in anything that has malt in it. So these dietary sugars are where we're going to be getting these other sugars that lead into glycolysis. So let's talk about the pathway itself. We're going to get into the nitty gritty of glycolysis. We're going to go through it step by step, looking at each individual step and what is happening. So I want you guys to understand at a stepwise manner what's going on, but also as a whole, what the pathway is accomplishing. Glycolysis is said to be broken down into two phases. The first phase is actually an energy requiring phase. So we actually have to put a little bit of energy into glycolysis. And then the second phase is the energy releasing phase. And we're going to release more energy to offset that little bit in. It's kind of like lighting a match. You have to put a little bit of energy in. So you lit the match and then you start the fire. So at the end, we're going to get a lot of heat, but we had to put in that little bit to start. In the energy requiring phase, each molecule of glucose is going to produce 2 molecules of glyceraldehyde 3-phosphate. This is sometimes written as GAP. And I'm talking about this now because glyceraldehyde 3-phosphate, or GAP, is an important molecule, as you might have guessed, in other pathways. So it's important for glycolysis, but you're going to be seeing it again. So remember its name. For glycolysis, each of these glyceraldehyde 3-phosphate molecules, these GAP molecules, then go into the energy releasing or the payoff phase. And remember, our very end molecule, the molecule that we stop at for glycolysis, is pyruvate. So each GAP molecule becomes a pyruvate molecule. Here is the very, very general idea of a breakdown of glucose. For each glucose molecule, we actually have to consume 2 ATP. Remember, that is our currency. So we're consuming 2 energy molecules, right? 2 ATP molecules. But in the output, we produce 4 ATP and we make pyruvate. So the general output, the general net yield here is 2 ATP per molecule of glucose. So this is that very, very, very general idea. We're going to get 2 ATP as a net for our glycolysis reaction. The very first step of glycolysis as part of the energy requiring phase is to use ATP. So this is one of the, remember, we're going to use 2 ATP to start. So one of the first ATPs come in here. We're going to utilize an ATP. And we're going to phosphorylate our sugar. Here is glucose. I've drawn glucose. And while I do not require that you know the structure for each of these steps, I suggest that you do. The reason is because if you see the structures and you actually draw out the steps as I have done here, I think it helps you understand chemically the pathway better. So you can just remember that glucose is phosphorylated here, but by actually drawing it out and showing what the molecule looks like and how it is changed, I think it will help you understand the pathway better. So in Biochem 1, we talked about the general sugars and their layout. This is glucose. Remember, glucose has an anomeric carbon here. That's why the squiggly line is there, right? It has its squiggly OH in which it can be up or down. The OH groups that are set in place for glucose always go down, up, down. And this is at position 2, 3, and 4. Here is position 5, and here is position 6, this point right here. So the OH group on position 6 gets phosphorylated. We've seen this type of reaction. You've seen this in the past, hopefully in Biochem 1, where we've looked at phosphorylation of carbohydrates. I've drawn the phosphate group as a P here, just as a simple reminder, but remember that phosphate groups look like this. They have a double bond oxygen, 2 negatively charged oxygens, and then this guy is what is actually attached. That's this oxygen right here. So we phosphorylated at the 6th position of glucose. That's why we make glucose 6-phosphate. See, the name makes sense when you draw it out. So again, first step, utilize 1 ATP, phosphorylate at the 6th position of glucose. So we're putting a phosphate on. And this enzyme, the actual enzyme that catalyzes this reaction, allows this reaction to happen. And spoilers, there's an enzyme for every step that we're going to talk about, is called hexokinase. So there is a special enzyme, special protein just to do this one step, which is phosphorylating glucose. Hexokinase. Kinases, any enzyme that has the word kinase at the end, means that it is putting a phosphate group on. And hexo, we have a 6-membered sugar. So that's how you can remember this is 6-membered, this is glucose, and it is phosphorylating kinase. What is the reason for this step? Why are we phosphorylating glucose in the first place? The rationale of the very first step of glycolysis is actually to move glucose. The addition of a phosphate traps glucose inside the cell. It gives glucose a charge. Remember, as I just showed here, phosphates have negative charges on them, right? So it charges the glucose, and this traps it in the cell. It can no longer move out of the lipid membrane of the cell freely, and it is now trapped. We want that because we want to break it down inside the cell, right? We want it to stay in the cytosol so that it can be broken down. It also is going to help lower the intracellular glucose concentration, because we've now converted glucose into glucose 6-phosphate, right? So what the cell sees is, I used to have 2 glucoses, but now I have 2 glucose 6-phosphates. Well, there's not enough glucose inside the cell, and it will start to bring more glucose in. As the glucose gets transformed to the cell, it looks like there is less glucose around. And so it starts importing more. And again, if we need energy, if we require that energy is being utilized, we need more glucose in the cell so that we can trap it and break it down. So that is the rationale behind this first step. This process is highly thermodynamically favorable and also irreversible. So phosphorylating glucose is going to happen in a semi-spontaneous way, and essentially is irreversible to go back to glucose. So once glucose is transformed into glucose 6-phosphate, it is not going to be going back to being glucose. So there needs to be some regulation of this step, because obviously you don't always at all times want to be turning all of your glucose into glucose 6-phosphate and then start it on the roller coaster of glycolysis. So this enzyme does need to be regulated. The enzyme that does this transformation turns glucose into glucose 6-phosphate. And it's regulated via inhibition. Substrate inhibition. Remember that enzymes can be turned on and off. We talked about inhibition of enzymes. We can have competitive inhibition. We can have non-competitive inhibition. Hexokinase gets inhibited if it is bound to ATP, then it will be inhibited. So an ATP with magnesium is going to facilitate the inhibition process. Why does this make sense? Well, if you have a lot of currency around, you have a lot of ATP around, it will inhibit hexokinase. It'll stop glycolysis. It'll stop turning glucose into glucose 6-phosphate. And that's reasonable because we already have a lot of energy. If there's not a lot of energy, if there's not a lot of currency, if there's not a lot of ATP around, then hexokinase will continue on its merry way, turning glucose into glucose 6-phosphate and continuing the trend of making more energy. So we have a nice balance there. This is what you're going to see through this whole semester. This balance of using products to then control the beginnings of reactions. Because if you have enough product, then you want to stop the reaction. If you don't have enough product, you want the reaction to keep going. This is the key behind substrate inhibition and the key behind how most of the functions in our body work. Okay, step 2. Glucose 6-phosphate is converted into fructose 6-phosphate. And this is through the enzyme phosphohexoisomerase. Isomerase, it's making an isomer of the product. It's just rearranging it, right? So we've taken a molecule and we're rearranging it from this glucose to a fructose. You can see it goes from a 6-membered ring to a 5-membered ring. It still has the same number of carbons. You see 1, 2, 3, 4, 5, 6 carbons. So they've just been rearranged so that the ring is kind of shortened and we've made another arm there. Why do we want to turn glucose 6-phosphate into fructose 6-phosphate? The reason is so that when we are going to cleave glucose, remember we're going to cleave it into two molecules that are 3 carbons long, it actually has a symmetric cleave. And fructose in general, just because of its molecular structure, happens to be easier to phosphorylate, which is what is also happening in this next step, excuse me. You're going to see we're going to do a second phosphorylation, and fructose is easier to phosphorylate. This step, when we're talking about the thermodynamics, is unfavorable and pretty reversible, meaning that glucose 6-phosphate and fructose 6-phosphate can kind of interconvert. And you see I've actually drawn the arrows specifically to show that these two can go between each other fairly easily. Unlike the step where we used ATP, steps that utilize ATP like this usually are going to be very, very, very strongly driven in one direction and essentially irreversible. Okay, step 3. As I hinted in the past, we want to phosphorylate that fructose. This is the second ATP we use in our energy utilization phase, right? This is still in the phase where we're using energy to build up our molecule so that when we break it down, it's going to release even more energy. So we're going to utilize our second ATP. It turns into ADP because it's giving away a phosphate. And it is going to phosphorylate the other side of that fructose. Notice that we made this nice other OH group that's hanging off, that's branched off from the fructose when we converted it into fructose. That OH group is now going to be the site of phosphorylation. It is in the 1 position because if we count, here is position 1, 2, 3, 4, 5, 6. So we're going to make fructose 1,6-bisphosphate, meaning that there are 2 phosphates and they are on position 1 and position 6. The enzyme that catalyzes this reaction is another very important enzyme. It is called phosphofructokinase 1. Whew. Okay, so let's again break down and think about the name. Remember, I said kinases always are going to be adding phosphates. So remember, kinases use ATP and they add phosphates on the molecules. And what are we adding a phosphate onto? Well, we're adding it onto a fructose that already had a phosphate. So that is why we get phosphofructokinase. What is the reason for this step? The rationale of step 3 is further activation of glucose. Remember, I said that we are putting energy in, we're putting a small amount of energy in, we're adding those phosphates on, and the reason we're doing it is because those phosphates are going to help us release a lot of energy in our energy releasing phase. So we're still further activating glucose. Similarly, I noted that we wanted to make our molecule symmetric, and the position of the phosphates also is symmetric, so that when we split this molecule, you're going to have a phosphate on each molecule that gets split. Just like with hexokinase, this step is highly thermodynamically favorable and irreversible. So the addition of that ATP makes this a very irreversible step. This is going just in that one direction. And it is also highly regulated. Hexokinase is fairly regulated. Phosphofructokinase 1 is it's one of the most regulated steps of glycolysis, and it is regulated by ATP. Again, for similar reasons. If we have a lot of ATP around, it will stop this step from happening because we do not need to continue down the irreversible stage of breaking down glucose if we have a lot of currency already. So ATP itself is going to, if there is a lot around, inhibit this enzyme from continuing. Some other inhibitory molecules. Fructose 2,6-biphosphate, which is slightly different than what we're using, fructose 1,6-biphosphate, is also a molecule that acts as an inhibitory molecule. There are also other metabolites. This is something we're going to get into further in this discussion. There are lots of molecules that are acting as regulatory and inhibitory molecules, either to activate or inactivate this step of glycolysis. Again, this step is kind of like it's the edge of the waterfall, right? It's an irreversible step. Once you go past this step, essentially you've committed to doing the breakdown. So this step is the reason that this is so highly regulated is if you have too much energy, you do not want to be wasting more ATP to continue down this step to further do glycolysis. That's why it's so highly regulated. Note that this, and like many other enzymes, also uses magnesium. Magnesium is an important cofactor in many of these enzymes. Okay, so we're still in the energy requiring phase, but now we are going to see the breakdown starts to occur, the actual cleavage of our molecule. Step 4 takes fructose 1,6 biphosphate and it lyses it. This is where the name lysis comes from in glycolysis, lysis, right? Glyco is glucose, lysis to break, right? So this is the lysing step. This step forms two 3-carbon molecules. It is going to form two different 3-carbon molecules. And you can actually see I've drawn them out here. So when we split this molecule in half, it splits pretty evenly, but we have a slight difference. Notice that here we have an OH group and here we have an aldehyde group. This is a carbonyl group at the end of a molecule. And here we have a ketone carbonyl group, and here we have an OH group. So they've kind of flipped positions, right? The one on the top is our glyceraldehyde 3-phosphate. And I noted that we're going to end up making two of these gap molecules. They are important. So this is glyceraldehyde 3-phosphate. Notice aldehyde. Here is the aldehyde group. It's at the very end of the molecule. And it has a phosphate on it, of course, on its third position. So it is a 3-carbon long 1, 2, 3-carbon molecule with an aldehyde group at the end and a phosphate on the other end. The other molecule that is made is called dihydroxyacetone phosphate. Sometimes this is also abbreviated dihydroxyacetone phosphate. There's not really a good way to say that. DHAP, I guess, DHAP. This molecule is temporarily around. Still important to remember it and that it exists because, again, we are going to be seeing it. It is not as important and as utilized as GAP is, but important note that it is made during this stage. We make two uneven molecules, GAP and DHAP. The rationale behind step 4 is just straightforwardly to cleave the sugar into its 3-carbon sugars. This is our cleavage stage to get ready to further break down those 3-carbon sugars. The reverse of this reaction, if you guys want to go all the way back to organic chemistry, is actually an aldol condensation. So taking a aldehyde and a ketone like this and condensing them into a ring, the reverse, is called an aldol condensation reaction. The reaction itself is unfavorable and reversible. So this can go back and forth in between making a ring and the two three-membered sugars. But the way that we make this so that it does not reverse, so that does not go back into the 3-membered sugar, is by keeping GAP concentrations low. GAP, like I said, is utilized in other reactions. So once it gets made here, it is then further utilized in other areas. And by keeping it low, there's less of a chance of it recombining with DHAP and then going backwards to form the fructose 1,6 bisphosphate. The enzyme that catalyzes this reaction is called aldolase. Okay, step 5. Step 5 is going to convert dihydroxyacetone phosphate into a second GAP molecule. This is where I said before that GAP is actually the molecule that gets utilized, and DHAP, dihydroxyacetone acetone phosphate is not the one that is utilized in these further steps of glycolysis. So we actually have to convert it into a GAP molecule during this step. So at this point, remember that we have made one dihydroxyacetone phosphate and one GAP. So the one GAP molecule that got made is just hanging out during this phase. The one dihydroxyacetone phosphate that is made is then isomerized into a GAP molecule using this triose phosphate isomerase. Again, it's making an isomer, right? We're just rearranging the molecule. We're not breaking anything off. We're just rearranging it. So you can see we're kind of flipping this carbonyl and OH group during this step. This is going to allow glycolysis to proceed further via one pathway. So we're just using GAP. We're not going to try to break down those molecules individually. The actual process is pretty reversible. Most of these isomerase processes where you're just rearranging molecules are going to be pretty reversible. And again, the way that this is allowed to continue forward, just like I talked about before, is by keeping those GAP concentrations low so that we don't spontaneously convert back into the dihydroxyacetone phosphate. We keep GAP concentrations fairly low. So it's more likely to be pulled towards creating GAP instead of the reverse. This step completes our preparatory phase. This is the end of the energy requiring phase. So we double phosphorylated, we put in those phosphates, and we actually cleave glucose into two GAP, and we've now made two GAP molecules. Having the two GAP molecules is the end of our energy requiring phase. And now we are going to move into the energy releasing phase. So I want to remind you from now on that each of these steps is essentially doubled. I'm showing you one GAP molecule here, right? Here is GAP, glyceraldehyde phosphate, 3-phosphate. But remember that we have two of them. So all of these steps are doubled. Step 6 is going to take our glyceraldehyde 3-phosphate and oxidize it. And it's going to be phosphorylated by inorganic phosphate, not by ATP. So we are going to add a phosphate on, but notice that the key thing here is that ATP isn't utilized. Instead, we're just taking a regular phosphate, that's inorganic phosphate, and adding it onto the molecule. This is going to form 1,3-bisphosphoglycerate. This is GAPDH, as we call it, another very important molecule that, again, you're going to see brought up through this semester. So we took our GAP molecule and we then phosphorylated it further, not with ATP, very important, by using NAD+, another currency molecule, converting it into NADH, and adding on a phosphate on the other end. So in this step, I want to break down some things. NADH is a molecule you are going to be seeing when we're talking about oxidation and reduction. Converting an NAD+ molecule into an NADH molecule requires the further oxidation of an adjacent molecule. And it acts to then carry electrons. So what's happening here is an NAD+ is picking up an electron to make an NADH. So remember that these NADH molecules are electron carrying. They have an electron. And when they dump off that electron, they become an NAD plus molecule. So in this step, we're actually, by giving the NADH an electron to carry, it is then going to be able to utilize that electron for energy way further on at the end of this story in our energy generation story and breakdown story. But just remember that NADH is important in these oxidation reactions because of its ability to carry electrons. So here, I'm just reminding you that there are two of all of these that are happening. Remember, we have two gaps. That means technically for a series of glycolysis, we had to put in two inorganic phosphates, two NAD pluses. We made two NADHs, and we generated two gap DHs, glyceraldehyde 3-phosphate or glyceraldehyde phosphate molecules here. What is the reasoning behind this further phosphorylation, but using our NADH? Again, adding on those phosphates is generating a high on energy compound, which is going to release its energy right at the end for our payoff to give us energy. This incorporates inorganic phosphate and generally is going to eventually allow for the production of ATP. So we're taking that phosphate not from an ATP, we're taking an inorganic phosphate, and it's going to help us make ATP in the end. This is technically the first energy yielding step of glycolysis, and it's through the formation of NADH that we are making energy. So energy through NADH should not be ignored. We talk about ATP a lot as this main currency of energy, but NADH essentially in the end is going to help us generate even more ATP. So it is also an energy yielding step by making NADH. This particular step utilizes this enzyme, as I noted, GAPDH, glyceraldehyde 3-phosphate dehydrogenase. Again, a very important enzyme that has an active site that is a cysteine. And this active site is very subjected to oxidative stress because of that thioester intermediate. I'm going to show you this in a second. But just note that GAPDH is very sensitive when you have oxidative stress. It is a reaction that is an oxidative process. So there is a redox reaction going on here. So it makes sense that this particular enzyme is sensitive to the oxidative environment. This step is also thermodynamically unfavorable, and it is actually reversible. So it is important that it gets coupled to the step 7, which we'll go into, to pull it along. So even though this is a nice energy generation step, it is one that can be reversible. But again, there are steps put in place through this process to continue pulling the reactions along, even though this can technically reverse. So I just quickly want to look at the actual enzyme and its kind of pocket that it utilizes. Glyceraldehyde 3-phosphate dehydrogenase, GAPDH, is again a very important enzyme that is very key to energy generation and glycolysis. So I wanted to note kind of how it works. So here is the pocket of glyceraldehyde 3-phosphate dehydrogenase, and you can see it utilizes a cysteine. Remember that these enzymes are a large corporation, large folded conglomerate of amino acids. So this is just one amino acid along its chain, but it happens to be an amino acid that is key to the binding pocket of this reaction. And you can see in step 2 here, we're going to bind our gap molecule here, and it is actually held in place by that cysteine, the sulfur on that cysteine in the pocket of this enzyme. And our NAD plus molecule is hanging out around in the enzyme. The rearrangement of the molecule is going to cause, you can see as our electron movement from this oxygen through this carbon and out with this hydrogen. So we're losing a hydrogen, but also an electron to the NAD plus, generating NADH. So the H, we do pull off a proton, we pull off a hydrogen, but with it comes an electron. So that is the electron carrying part of the NAD plus. So the enzyme substrate intermediate is thus oxidized by the NAD plus, which is bound into the active site. So you have this oxidation. Remember, oxidation is loss of an electron. So our actual molecule, gap molecule, got oxidized. It lost an electron to our NAD plus to make NADH. The NADH then is going to leave and is replaced by a new one, which is regenerated. And you have then this phosphate come in our final portion, which then can attack and attach to that carbonyl, forming our final phosphorylated molecule. So it is through the use of this NAD plus that we actually have a rearrangement that allows for the phosphate to bind. This reaction, again, is using this essential cysteine residue, and it can be inhibited through heavy metals. So things like, for example, mercury can actually irreversibly inhibit this enzyme. This is brought up in relation to some of the effects of mercury poisoning. So reactions in which heavy metals can bind to this particular cysteine will stop the gap DH from continuing and essentially halt glycolysis and most energy production in cells. Okay, so let's look at step 7. We have now made 2 of our 1,3-bisphosphoglycerates. This is a gap molecule that has been phosphorylated on each side, right? Called 1,3-bisphosphoglycerate. This molecule is now going to start breaking down and releasing ATP. So in step 7, we are going to see our first production of ATP. And I'm going to remind you again that all of these steps are doubled because we have 2 of these 3 carbon molecules. This uses phosphoglycerate kinase. Kinase deals with ATP and phosphorylation. Remember, that is a key to remind you that those are involved. And we're going to essentially break off one of our phosphates and take an ADP and make ATP. Again, Step 7 is doubled. So you actually took 2 ADPs and made 2 ATPs with each of those molecules. This is very generally the rationale to use phosphorylation to make ATP. Remember that kinases are enzymes that transfer phosphate between ATPs and various substrates, as just noted. And so this is also using a kinase, even though in this step we're doing it in the reverse, right? We're taking an ADP and making it an ATP. Still, generally the idea of transferring phosphate on ATPs. This step is highly favorable and also reversible. So note that in the step in which we are producing ATP, we're actually making an energy molecule. Building an energy molecule is going to be reversible. Breakdown of energy molecules are going to be irreversible. And this is reversible due to its coupling with that gap DH reaction. Our previous reaction, unfavorable and reversible. This one is favorable reaction, but the reversibility of it is going to be tied to that gap DH reaction. So these reactions coupled together ultimately make this reversible, but as I said, ultimately favorable. If you see, our delta G is actually a negative delta G in this. Step 8. Step 8 is another kind of rearrangement reaction. We're going to transform our 3-phosphoglycerate into 2 phosphoglycerates. We're actually just moving a phosphate, and this is using phosphoglycerate mutase. What is the reasoning for this? This is to ultimately form a high energy phosphate compound. And it's using this mutate to actually migrate the functional group. One of the active sites on this mutase is a histidine, and it is modified to a phosphohistidine, in fact. So you can see this is going to temporarily modify that histidine to a phosphohistidine by taking the phosphate, moving it onto the histidine, and then you have the movement between where we're taking the phosphate off and moving it onto the other position, on position 2 instead of position 3. So we're kind of like moving a phosphate on and off of a histidine, making a phosphohistidine in between. This reaction is unfavorable and reversible. And again, the reason that it is able to continue is because the reactant concentration, that's the concentration of the 3-phosphoglycerate, is kept high to push forward. Two phosphoglycerate in step 9, coming to the end here, is then dehydrated. This is our first and only dehydration step along the process. And it is going to form phosphophenol pyruvate. Again, this molecule is one I'm going to note. This is the penultimate molecule, the molecule right before we get to our last step, but it also is going to be a molecule that you see pop up in other pathways. So here we are taking enolase and dehydrating this alcohol to form a double bond and form phosphoenolpyruvate. So you can see that it's important to get to this stage where we can do this dehydration, that we move that phosphate to the 2 position in the previous step. Again, rationale here is we're just building to a further high energy compound, which you will see in the end will allow us to finally make our pyruvate in our very last step. 2-phosphoglycerate is not a very good phosphate donor. And so by dehydrating it to make this this phosphophenolpyruvate compound, we actually get a better donor of phosphate. This reaction is, again, slightly unfavorable and reversible between the two. But what's keeping it pulling forward is the product concentration that is altered to be kept low to pull forward. Notice the trend when we have reversible steps. Mainly what happens is that we utilize those products so quickly. Those products are utilized in many areas so that they are kept low so that we can continue pulling the reaction forward. Okay, last step of glycolysis. Pyruvate is formed. So definitely a key molecule to both know the name of and know what it looks like. Here is pyruvate. You can see it is a 3-carbon molecule that has these 2 carbonyl groups on it and this O- group. This sometimes can have a hydrogen on it, an OH group. So essentially like a carboxylic acid here. So we're taking phosphoenolpyruvate and finally removing that very last phosphate. This is going to use the enzyme pyruvate kinase. Again, a very important enzyme for the final step. And it is an enzyme that is, again, a kinase because we are taking pyruvate and, we're taking a phosphate and attaching it to ADP to make ATP. And again, I'm going to remind you that all of these steps are doubled. So we technically took 2 phosphophenolpyruvates, 2 ADPs, and 2 ATPs to make 2 pyruvates at the very end. This is yet again another substrate level phosphorylation making ATP, and it helps us net produce those 2 ATPs per glucose. The loss of this phosphate on the phosphoenolpyruvate, the one that we saw earlier, phosphophenolpyruvate right here, is going to actually allow, when we form pyruvate, this phenylenol tautomerization, ketoenol tautomerization. Had a lot of enol words in there, phenol, So it is a ketone enol tautomerization, meaning going between an enol form and a keto form spontaneously. Pyruvate can undergo this. You can see here is the enol form and the keto form. The keto form is the form that was shown here, which is the most prominent form that you usually see of this molecule. But as I said, it can undergo tautomerization to form an enol form. Tautomerization is just the movement of electrons spontaneously within a molecule. And you can see here we're moving the electrons from this double bond oxygen here, carbonyl, to this bond between carbon and methyl group. And we also have the migration of a hydrogen. The reason this is important to talk about that pyruvate undergoes this is because it is one way that effectively lowers the concentration of the product. So it will help drive the reaction to continue to form more pyruvate, because if pyruvate is in the enol form versus the keto form, effectively it looks like there is not as much pyruvate around, and so the reaction will continue to form pyruvate. So this ability of pyruvate to go to tautomerize between its enol and keto form is actually important for the progress of breaking down glucose via glycolysis. This enzyme that actually forms pyruvate, again, is a kinase. It's heavily reliant on magnesium or manganese for its activity. So it's very important that nutritionally magnesium, as I've noted throughout this, is available for glycolysis to occur. So one thing you might think about that is if you have magnesium deficiency, you will actually see problems in glycolysis and breakdown, and just like general energy production. Again, highly thermodynamically favorable and irreversible for this kinase reaction. And in addition to the thermodynamic favorability and irreversibility, we have this the tautomerization that also pulls along the reaction to continue towards forming pyruvate. This step is also a step that is regulated. The enzyme is regulated. It is regulated by ATP. It's also regulated by other divalent metals and other metabolites. So yet again, if you want to think about regulation of ATP here, of course, we don't want to continue to make more ATP if we already have a lot of ATP. And so ATP in this case will also stop the continuance, or it will inhibit pyruvate kinase from making more pyruvate. Okay, so let's look at a review. I know that was a lot, and I wanted to go through each of the steps and kind of talk about what is happening at each of them. Ultimately, what is used? We take one glucose, we make a net of 2 ATP. Excuse me, we take one glucose, we use 2 ATP, and we use 2 NAD+. These are the things that are going into the reaction. These are the main molecules we want to talk about that go into the reaction. What do we make from the reaction? We make 2 pyruvate, right? Our glucose breaks down into 2 molecules that are pyruvate. This pyruvate can go on into different areas. We make 4 ATP. Remember, there are 2 steps at the end that generate ATP, and each of them are doubled. And we make 2 NADH. So ultimately here, we can write out this reaction as glucose 2 NAD+. We use 2 ADP and 2 inorganic phosphates. So these phosphates are going to ultimately be used to make the ATP. We generate 2 pyruvate, 2 NADHs, 2 protons, and 2 ATPs. These protons are connected to the generation of the NADH through NAD+. So this is our net reaction. I'm reminding you it's a net reaction because, as you can see up here, we did use 2 ATP, but we made 4 ATP. The net is that we made 2 ATP, right? This is the net amount of ATP we made because 2 were used and 4 were made. So when we're writing our net reaction out, you can see we're going to take 2 ATP and make 2 ATP. That is the net amount of ATP we used. We need to add 2 phosphates to our ADP to make the ATP. And again, 2 NADHs are going, NAD pluses, excuse me, are going in to make 2 NADHs. Another point of review I want to remind you of is that glycolysis is heavily regulated. I tried to stop at various points to talk about enzymes that are regulation points along the pathway. I'm going to highlight those again more, but those enzymes are important to remember. It's important to understand those regulation points because they are there so that the production of ATP is only occurring when needed, right? We do not want to waste energy to make ATP if it is not needed. So we're going to talk a little bit about the fate of pyruvate right now. Once you finish glycolysis and you've made this energy, you've made your ATP and you've made your NADH, you now have 2 pyruvates. And those pyruvates, as noted, can go on into further reactions. One of those reactions we're going to be talking a lot about eventually is going into the citric acid, or TCA, tricarboxylic acid cycle. This is one reaction that many mammalian organisms, or just in general, many organisms use, is the citric acid cycle. But this requires oxygen. Glycolysis does not require oxygen, but further breakdown of pyruvate in aerobic conditions using oxygen can occur in this citric acid cycle. But what about in situations where you want to further break down pyruvate, or you need further energy, but you do not have oxygen? You are an anaerobic condition. Well, this occurs in fermentation. This can be the second route of pyruvate to form lactate or lactic acid via fermentation. And again, this is when we are still needing energy, but we are in low oxygen conditions. Fermentation can occur via 2 types of routes. One is maybe one you're familiar with, in which you have anaerobic organisms that can ferment pyruvate into ethanol and CO2. These are how yeast works, for example. Fermentation into ethanol is a pathway that pyruvate can take. Again, this is a pathway that only some organisms can utilize. This is going to be organisms like yeast and certain bacteria, which can, in no oxygen or low oxygen environments, make ethanol and CO2 from pyruvate. In other organisms like us, humans, we cannot do this. We do not just spontaneously make ethanol. We don't get ourselves drunk spontaneously. We still do have a pathway of fermentation or of breakdown of pyruvate in anaerobic or low oxygen conditions. And this is turning it into lactate. So these are going to be two pathways we'll talk about, slightly different, but there are similarities of anaerobic breakdown of pyruvate. So again, these are conditions in which you do not have oxygen. If you do have an oxygenated environment, then the pathway that will be utilized, for example, in humans, is through this pathway of anaerobic conditions of making, using the citric acid cycle. Let's talk about lactic acid fermentation. Lactic acid fermentation takes pyruvate and again converts it into lactate. And by doing this, it is going to utilize that NADH that we just made in glycolysis. And it's going to make an NAD+. So notice this is actually the reduction of our molecule. This is going to occur through lactate dehydrogenase. Very important enzyme that we're going to talk about. This is an enzyme that is utilized in humans to make lactate from pyruvate. It's also abbreviated as LDH. LDH is actually measured in a lot of medical settings because it is a marker. How much lactate dehydrogenase is being used is a marker for how your tissues are breaking down pyruvate, if they are stressed, if they are in low oxygen versus high oxygen environment. So at a tissue level, right, tissues in our body, if they become depleted of oxygen, if they're working very hard, they will start to utilize lactate dehydrogenase to make lactate out of pyruvate. Why would we do this? I just showed you what the reaction is. Using lactate dehydrogenase, we take pyruvate, we make lactate. But why? Why is this going to be efficient in energy generation? And actually, if you remember, I actually said that NADH is an energy molecule. So why are we taking an energy molecule and then breaking it back into NAD+? Important questions. In animals, when we cannot be supplied with sufficient oxygen to support normal aerobic oxidation of pyruvate, so that's citric acid cycle, if we cannot go down that pathway, then the pathway we have to continue using is actually glycolysis. But glycolysis uses NAD+ and makes NADH. So if you want to continue glycolysis, if you want to continue making ATP and energy in glycolysis, you actually have to start regenerating that NAD+ so that it can be used in glycolysis. So the whole point of lactic acid fermentation in animals is just so that we can regenerate this NAD+, so it can cycle back into glycolysis so that we can continue breaking down glucose and making ATP through glycolysis. This is going to happen a lot in animals during strenuous exercise. So when you're using your muscles a lot and you're using them, they're going to quickly start burning oxygen, using oxygen, and you're breathing heavily and you're trying to get oxygen to your muscles quicker through blood movement, but you might start to have areas where there is low oxygenation because you're just using the oxygen so fast. So what happens is your muscles are going to switch over to just using glycolysis. They don't have enough oxygen around to use citric acid cycle, which we need oxygen for that. And so they still need energy. They're still working hard. They still need to make ATP. We're going to switch to only using glycolysis. And to do that, I need more NAD plus. All that NADH I just made needs to be reconverted into NAD plus so that I can keep doing glycolysis. So what happens is you actually will switch to fermentation, lactic acid fermentation. You'll make that NAD plus you need. You'll continue doing glycolysis. You'll continue making ATP, but all of that pyruvate that you made will now be formed into lactate or lactic acid. You've probably heard of this, lactic acid buildup in muscles, right? When this occurs, when we have a low oxygen environment and we switch over to this fermentation, you will be able to make energy, not as efficiently, but you'll be able to make it fast with glycolysis. But the side product is you make lactic acid. And this is what people describe as like a burning sensation when you have a lot of lactic acid buildup in your muscles. So some tissues and cells, such as erythrocytes, this is actually your heart muscles, they do not have mitochondria and they can't do citric acid cycle. So they actually rely completely on this lactic acid fermentation and doing glycolysis. So they produce lactate from glucose even when they have oxygen. And this is actually, this note I just made is very, very interesting and very key. So heart cells only do this glycolysis and lactic acid fermentation. So they are one of the cells in the bodies that have the highest production of LDH, this lactic acid dehydrogenase enzyme, because they need it so they can do fermentation. So this muscle is this is a muscle in your body that has the highest amount of LDH. And as I noted, LDH is a measurement of how your heart health is. So this is actually a lab test you can go get done. If you go to the doctors, they'll measure your LDH and it is related to your heart health. Okay, let's talk about ethanol fermentation now. Remember that I said some microorganisms can also do fermentation. They are anaerobic organisms. They do not use the citric acid cycle. They don't even have the capability to use the citric acid cycle. They use these low oxygen fermentation to continue getting energy, but theirs is slightly different than in humans. They do not do lactic acid fermentation. They do ethanol fermentation. This is actually a two-step process and is very key to yeast. So hint for those that are in the lab where we're going to be doing sourdough cultures and looking at yeast, this is the main reaction that those cultures utilize. There are, again, two steps. One uses pyruvate decarboxylase. So we actually take pyruvate and break off a carbon in the form of CO2. So it goes from a 3-carbon molecule, you can see, and then it actually breaks off this whole portion here, along with, in doing this, it takes, you can see 2 oxygens and a carbon, so we make CO2, leaving behind this 2-carbon molecule. And this is an aldehyde, remember? We then take this aldehyde and then do a final step to make a ethanol. Ethanol, again, is a 2-carbon long molecule with an OH at the end, right? This is ethanol. The second step, alcohol dehydrogenase, is the most important step. It is this ability to convert alcohol into this aldehyde and vice versa. And it takes NADH and makes NAD+. Remember, again, this is the key part of these steps because we are regenerating NAD+ to feed it back into glycolysis. So these microorganisms are using the formation of ethanol to make their NAD+ to then continue breaking down glucose and getting energy. This is actually things like yeast and certain bacteria are completely reliant on this simple steps of glycolysis followed by fermentation to just keep their power going to make ATP. So this is the only way. This is many anaerobic organisms. They don't use oxygen at all. They just use these fermentation pathways and glycolysis to get their energy. So again, this is for microorganisms in ethanol fermentation is two-step reaction of taking pyruvate to then in the very end form ethanol, and it is irreversible. As humans, we don't have pyruvate decarboxylase. That is the first enzyme, this enzyme that takes pyruvate and makes our aldehyde. But we actually do have alcohol dehydrogenase. We have alcohol dehydrogenase, but we use it in the opposite way. We actually use it to take alcohol and convert it into other molecules. So important note that while we don't have this first molecule, we can't ultimately form ethanol. We can use that second enzyme, alcohol dehydrogenase, to metabolize ethanol in our bodies. So interesting note, we only have half of those. Humans, again, are going to rely on lactic acid fermentation using LDH, remember, to do our fermentation for pyruvate. This ethanol fermentation is very important still in our lives because microorganisms that use ethanol fermentation are responsible for the beer that we drink and the bread that we eat. Yeast in particular is utilized for its ethanol fermentation for many reasons. If you note, the first step, which is pyruvate decarboxylase, makes CO2. And actually, that's where all of this carbonation comes from when you drink beer or other drinks that are carbonated using yeast. It's from pyruvate decarboxylase, the release of that CO2. This is actually also how dough rises when you're baking bread. The activation of those yeast cells in making, in consuming glucose and then breaking it down further through pyruvate decarboxylase releases CO2 and puffs up the bread and rises it. So this CO2 production, the step in CO2 production is very important, and again is the reason that we see carbonation and dough rising when we utilize yeast. Now, both of these steps are also regulated. They do require cofactors. Pyruvate decarboxylase uses magnesium. Again, magnesium is very important in general in both glycolysis and fermentation. It also uses thiamine phosphate. This is a cofactor we'll see again in the future. And alcohol dehydrogenase actually utilizes, of course, NAD+ because it is taking, it is making that NAD+ from NADH. And also zinc. So zinc is an important cofactor in alcohol dehydrogenase. So let's talk about a little bit on the application note in looking at glycolysis and cancer. The anaerobic metabolism of glucose in tumor cells is often used. So this process of using glycolysis and fermentation. This is anaerobic metabolism. I'm going to key in on that again. That means metabolism when there's no or low oxygen, right? It does yield less ATP than if we continue this down the citric acid cycle. Again, we're going to actually look at and calculate how much ATP you would get from that. But you only get 2 ATP for every glucose that you break down. Whereas if you completely break that pyruvate down in oxygenated or aerobic conditions, you actually get 30 ATP out of each glucose. So it gives you far less energy. It is faster, which is why, for example, if you're utilizing your muscles and you're in low oxygen, it gives you energy fast, whereas these aerobic conditions to further break down pyruvate are not as fast. They're a much slower process. But you can accomplish just the glycolysis and formation of 2 ATP, again, in anaerobic conditions, no oxygen. So because of this, tumor cells actually are very hungry because they only do anaerobic metabolism, or they rely mostly on anaerobic metabolism because they are a low oxygen environment. They end up consuming much more glucose to get ATP than most cells. So glucose transporters and most of the enzymes involved in glycolysis are actually usually overproduced in tumors. You'll see them as markers a lot. Anything that you need to get glucose into the cell or to break it down in glycolysis are usually way, way, way overproduced in cancer, because cancer is so reliant on this glycolysis pathway to get its energy. And it has to be doing it all the time and more often than most cells. So you can see here is ATP synthesis that is in conditions of what we call normoxia. This is normal oxygen conditions versus hypoxia, where we have abnormal or low oxygen conditions. Normal differentiated tissues, right? We can go through the process of breaking down glucose through 2 main pathways. And we've already talked about this. We can do glucose into pyruvate. This is glycolysis. And then that pyruvate can go down aerobic conditions to make lots of energy, or it can go down anaerobic conditions to make lactate for a smaller amount of energy, but faster. Tumors, in contrast, only utilize generally this pathway in which we are forming lactate. A very small percent of these cells are actually doing the aerobic glycolysis, in which they are making that energy through the citric acid cycle. This actually has a name to it. So if you're ever looking at papers or researching, it's called the Warburg effect. This is that even in situations where tumors have oxygen, they are still reliant almost solely on the glycolysis and formation of lactate to get their energy. One effect of this, the Warburg effect, one thing that you'll note is because tumors are so reliant on forming lactate, remember that this is also called lactic acid. So it is an acid, and that means that tumor pHs are very low. That's because they're forming so much acid because they are just stuck on this formation of doing glycolysis in tandem with fermentation, lactic acid fermentation. All right, well, that is the end of our first chapter looking at glycolysis.